Okay. This is just our title. I'm going to tell you a little bit about a aquatic invasive species of freshwater jellyfish, collaborating with a colleague at Kansas, University of Kansas. Next slide. So this is a lot on here, but these are our goals and objectives. So let me just break these down quickly. We're looking at the biology of this organism, and we want to determine the ecological impact it may have in aquatic systems, perhaps due to climate change. Temperature is one of the many factors that's involved in the life cycle. So what environmental cues are out there that perhaps may lead to the medusa stage or the jellyfish stage? A lot of people don't know there's a freshwater jellyfish, but there is. And that's part of what we're excited about. Use the cues to predict where and when this might occur in South Lake, Michigan areas. Replicate the conditions in the lab. So we've got some lab work we're working on to produce or induce the medusa or the jellyfish formation and hopefully get those to reproductive state. Determine the genetic mechanisms involved in the blooms and also perhaps aspects of the life cycle and compare that to marine systems. And lastly, scientific literacy on invasive aquatic species, developing educational models, which we've been doing that. It's pretty exciting. So I'm excited to talk to some of you about educational things and getting the public involved. Next slide. So these are just a schematic of the picture. I've put in red our main focus. My colleague and I, we've been able to culture this organism in the lab. The polyp stage, they're small. So how big are these jellyfish? About the size of a quarter or a little bigger. You do see them, but the polyp stage is about 1 millimeter. And so they're out there typically, and we've been able to culture them through the asexual stage, the lower part, but it's that red area. We've both been able to get the medusa in the lab, but to sustain it and get it to produce gonads, to then get egg and sperm and complete that life cycle is what we're working on. So that's where our challenge is, and no one has been able to do that yet. So we're excited about hopefully getting that to happen. Next slide. This is just a series of pictures showing you the medusa that buds. If you start up there, the medusa buds from a polyp. So the polyp is just like Hydra, only it doesn't have tentacles, and it's about 1 millimeter, and it will bud the medusa or the little baby jellyfish on the side. And then you can see A, B, C, and then D are these little things that are about 1.5 millimeters. And then eventually they develop gonads, and we go from there. Next slide. So our research, the first 3 objectives, we've got field work that we've been doing, collecting water samples in terms of pH, temperature, a variety of things. We've got 3 sites cultured in the lab right now from Illinois, and we have 3 Indiana sites that I'm still processing, and after lunch I'm going to go to 2 more sites today. I've got permission to get in the water. We're also replicating this in artificial ponds in Kansas. This top picture here is my colleague, Dr. Paulin Cartwright, with her student, Kent. He's working on a master's, so a good bit of this work plugs into his master's work. So they've got an artificial pond there. In the lab, we both are culturing these. We're culturing different populations. We've been working on these for a bit. We both have a population. I have a Panama population. She has a Japanese population. We're sort of comparing that to these freshwater populations in the area. And so these are some pictures of my students. Sarah Chang worked with me in the summer. She's an education major with a lean into science. She's the one that's done a great job of developing the curricular material for us, which I'll mention. And then the genetic cues to signal these blooms. This is part of Kent's master's work, and he has already begun sequencing the genome and transcriptomes in the life cycle stages, and we want to look at some of the molecular aspects of the populations. Our last goal, next slide, is the scientific literacy and education. So we want to increase the awareness of invasive species and develop curricular material, and as I said, Sarah Chang did that this summer. And so we've got at least 5 to 8 days of classroom material. We've been working with some schools, and we'll start visiting those schools. We've got this 5-day curricular material plan. Paulin Cartwright presented to the Kansas Association of the Biology Teachers, and they were all pumped. And we've already got teachers wanting us to come and visit their schools, so we're probably going to get a little overwhelmed, but we're excited about that. And we'll modify that curricular program to meet the needs of the schools and the teachers. And we'll go to the schools and or follow that up with like Zoom calls and things like that with the classroom. And then we're going to create a website to have people log the sighting of these. And the last slide is simply an update on where we're at. So our field work, our website work, our lab work, undergraduates, I have 3, maybe 4 that are working with me. Paulin has a graduate student and he's doing the molecular work. And then our modules are developed and we just need to tweak those and modify them and interact with educators. So we could use help connecting in northern Illinois or Indiana areas for educational purposes as well. Thank you.